Hello everyone and welcome. In this video we're checking out variant 1 of October-November 2017. Before we start, make sure to check out the free notes I have available for you in the comments section down below. Now let's go ahead and start with question number 1. Which row is correct for a typical plant cell? Cell wall, cellulose, cell diameter 1 to 5, definitely not 1 to 5 micrometers. This is your typical bacterial cell. Ribosomes, both 70S and 80S. Okay, you're going to only find 70S ribosomes in bacteria. You're also going to find bacteria to be 1 to 5 micrometers in diameter, and peptidoglycan cell walls will be found in bacteria. So cellulose and 5 to 40 and 70S and 80S. An electron micrograph of a cell shows large quantities of rough endoplasmic reticulum and many Golgi bodies. What type of cell is being viewed? Is it bacterium? Obviously not bacterium, because bacterium have no organelles. So these guys have no membrane bound organelles, I think is the better wording for this. Yeah, no membrane bound organelles. Guard cell, I don't think guard cells have a bunch of Golgi bodies and rough endoplasmic reticulums. It's not really their function to synthesize a bunch of proteins more than opening and closing the stomata. So I would not choose guard cell. Lymphocyte could definitely be an answer because they synthesize antibodies. Antibodies are proteins, and who synthesizes proteins? Well, it's the rough endoplasmic reticulum, and who modifies them? It's the Golgi bodies. Therefore, I would definitely choose C, and for same reasons, I would not choose B. I would not choose D. Which cell structures can form vesicles? Okay, cell structures that can form vesicles, cell surface membrane, endoplasmic reticulum, Golgi body. Well, the endoplasmic reticulum can definitely form vesicles, and so does the cell surface membrane, and so do the Golgi body. All these structures can form vesicles because Golgi body, it basically it's one of its main functions is to produce secretory vesicles. Endoplasmic reticulum, it's made of it's basically a single membranous organelle that secretes proteins, and how these proteins are going to be released is going to be in vesicles, as well as lipids by the smooth endoplasmic reticulum. They're going to be released in vesicles. Cell surface membrane undergoes endocytosis and basically forms these vesicles as well, or pinocytosis, whatever. It also obviously makes vesicles. Which set of measurements is correct? Diameter of a capillary, the diameter of a capillary, diameter of a red blood cell, and the thickness of the cell surface membrane of a red blood cell. Now, obviously, the diameter of a capillary is going to be 7 micrometers, and so is the diameter of a red blood cell. And the thickness of the cell surface membrane of a red blood cell is actually 7 nanometers. Now, a thickness of any cell surface membrane, the average is going to be 7 nanometers, therefore A. The size of the measles virus is approximately 150 nanometers. The Mimi virus is approximately 4.5 times larger, which means it's exactly 4.5 times 150. That is 675 nanometers. Okay, it's pretty big. And then when it comes to the Pandora virus, it's approximately 1.5 times larger than the Mimi virus. Okay, so you multiply that by 765, so it's 1000 and 2.5. That's pretty big. Now, which viruses can be seen using both light microscope with a resolution of 0.25 micrometers and electron microscope? Now, the resolution in nanometers, if we could convert to nanometers, 0.25 times 1000 is going to be 250. Now, how you convert from micrometers to nanometers is by multiplying by 1000. So this is what I did here. So you get 250. Now, for you to be able to see anything using any kind of microscope, it has to be having a distance that's greater than the resolution. So, for example, the diameter of the measles virus should be greater than the resolution of 250 for it to be seen using the light microscope. The electron microscope has a very small resolution, and I'm pretty sure all of you guys are aware of that, of the fact that it's much smaller than the light microscope, and it's, to be exact, 0.1 nanometers, which means you can see anything bigger than 0.1 nanometers. So you can obviously see the Mimi and the measles virus as well, but no, obviously you can't see the measles virus in this case because the measles virus is less than 0.25 micrometers in diameter. So no, the only thing that will not be seen is going to be the measles virus. Everything else will be. So that's B. Okay, which carbohydrate gives a brick red color when boiled with Benedict's solution? Okay, brick red color when boiled with Benedict's, cellulose, fructose, glycogen, sucrose. So we're looking for a positive result. Now, the only thing that's going to give a positive result with Benedict's is a reducing sugar. Now, cellulose and glycogen are both non-reducing sugars because they're polymers. Sucrose is the only non-reducing disaccharide, and that's why it does not give a positive result with Benedict's. Lastly, fructose is a monosaccharide, and all monosaccharides are reducing sugars, therefore B is your answer. Which of two polysaccharides both have 1,6 glycosidic bonds and are branched? Okay, two polysaccharides which have both, like, branches and obviously 1,6 glycosidic bonds. You have to know that 1,6 glycosidic bonds mean branches because they only occur when branching happens. So basically them saying this is just basically enough for them to list branches. So even if like you don't see the branches, just the fact that they have 1,6 glycosidic bonds should indicate to you that they are branched. Anyway, is it amylopectin and amylose? Is it amylopectin and glycogen, amylose and glycogen, or glycogen and cellulose? Now, amylose is actually a non-branched polymer, and so the only polysaccharides here that have let's say the only polymers that you should know that are branched are amylopectin and glycogen. Amylose is not branched, it's just helical in shape, and cellulose is not branched, it's just straight in shape and it doesn't have any branches in it. So it's amylopectin and glycogen. 3 carbohydrate molecules are shown. So you have three monosaccharides. You see which two molecules combine to form a molecule of sucrose. Sucrose is made of one alpha glucose molecule and fructose. So which is alpha glucose here? Alpha glucose, in order for you to point out if it's alpha or beta glucose, you look at the hydroxyl groups, which are these OH groups. So if they're pointed down, down, up, down, that's alpha. Like this, down, down, up, down, that's alpha. But if they're pointed up, down, like here, up, down, that's beta. So it's going to be alpha glucose and fructose, which is basically this one. You don't really have to guess here. It's this one, because fructose is a 5-membered carbon chain. It is going to have hydroxyl groups in this order, and it's it's a hexose sugar, but it's still a 5-membered one. Okay, so which two molecules combine to form a molecule of sucrose? It's 1 and 2, which is A. Okay, moving on, you have the diagram shows amino acids in a polypeptide. So you have a bunch of amino acids here, and he says an enzyme catalyzes the hydrolysis of bonds between polar basic amino acids and non-polar amino acids. Interesting. So how many peptide chains of amino acids will be formed as a result of the hydrolysis of the polypeptide? How many chains of amino acids will be formed? Okay, so the ones here, you have a key. Okay, the ones that are polar basic are the dark square ones, and then the ones that are polar acidic are hexagonal in shape and they're light, and then polar is just hexagon and it's darker, and then non-polar is a spherical looking one. So he says here that an enzyme that catalyzes bonds specifically between polar basic and non-polar amino acids, which means we're looking for this one and this one. Okay, so any square and a circle. Okay, so how many peptide bonds will be formed, how many peptide chains will be formed as a result of this hydrolysis? So let's see where the hydrolysis happens here. Square, circle, the first one is here, the second one to be broken is right there, and then we also have another one here, and then another one right there. Okay, and then you also have one here, and that's it. So this leaves us with this one, and then 2, and then 3, and then 4 here, and then 5 and 6. So it's 1, 2, 3, 4, 5, 6 chains. So this should be B. Okay, a lysosome is an enzyme found in tears and saliva. Which statement refers only to the tertiary structure of lysozyme? Okay, lysozyme is an enzyme found in tears and saliva, and he asks about the tertiary structure of lysozyme. A section of molecule consists of an alpha helix. Alpha helix has to do with secondary structure and does not refer to tertiary structure. Therefore, we can cancel this. Okay, it contains a single polypeptide. A single polypeptide refers to the quaternary structure, because quaternary structure is all about the number of polypeptide chains that are available. And as soon as you have more than one polypeptide chain in an enzyme, this means it has a quaternary structure. So no, this does not refer to the tertiary structure. 129 amino acids long. This has to do more with the primary structure. The 35th and 52nd amino acid help form the active site. So yes, 35th and 52nd help form the active site. The active site is supposed to be this region that has a specific tertiary or 3D structure, which is going to be where enzymes or like substrates are going to bind. So the amino acids involved in this formation, they could have hydrogen bonds between them, they could have disulfide bonds between them, or hydrophobic interactions, or it could be ionic bonds. You never know. So that's why they listed amino acids. Now some students may look at this and get confused. It's like, is this referring to like primary structure? But literally it's the only answer that actually refers to tertiary structure, so D. Okay. Some RNA molecules called ribozymes can catalyze reactions in a similar way to protein enzymes. Most of these ribozymes have other RNA molecules as their substrates and catalyze reactions that break specific sugar phosphate bonds in the substrate molecules. So what they do is they break specific sugar phosphate bonds in substrate molecules. Which statements about these ribozymes are correct? Hydrogen, ionic, and disulfide bonds will be involved in the ribozyme structure. Are they involved in the ribozyme structure? Well, for what we know about ribozymes is that they are supposed to be made of RNA. RNA is supposed to be made of nucleotides. So nucleotides can form, yes, hydrogen bonding between one another. Can they form ionic bonds is basically... Okay, maybe they could form ionic bonds, or could they form disulfide interactions is the question. And disulfide interactions only happen between, we saw in amino acids, they happen between cysteine amino acids, which basically have the SH groups. Right, so what's it called? The SH, they're going to bond together from this covalent bond. Now, in the case of a nucleotide, they don't have any sulfur. So, no, disulfide bonds are not going to form. Maybe ionic bonds could form and hydrogen bonds. However, no, these are not all involved in the ribozyme structure. The active site of a ribozyme is formed from a specific sequence of nucleotides. Yes, of course, just like any RNA molecule is formed from a sequence of nucleotides. Ribozymes can form because RNA can have a specific secondary and tertiary structure. Now I know it's a bit confusing to you guys because we've just heard of tertiary, primary, secondary structures when it comes to proteins, but no, a bunch of molecules have both primary, secondary, and tertiary. These are just different levels of organization of any molecule. So yes, ribozymes, just like any enzyme, have secondary and tertiary, quaternary levels of organization because they want to have that, shape, that globular shape, and they want to be able to form these basically active sites that allow the substrates to bind to them, and therefore yes, it's 2 and 3 only. The graph shows the effect of an enzyme on a reaction. So you have the energy, you have the substrate, you have the product, and you could see how the enzyme is going to lower the activation energy, and this is how it basically speeds up the reaction. Which labels are correct? X is the reaction without the enzyme, and W is the reaction with the W is the activation energy. W is the change in activation energy with using enzyme versus without using enzyme. So no, W, this is wrong. Okay, is X the reaction without the enzyme? X is the dotted one, so yes, X is the reaction without the enzyme. X the reaction with the enzyme, so no, this is wrong. Z is the overall energy change. Yes, Z is in fact the overall enthalpy change or energy change. Y is the reaction with the enzyme, whereas Y is the reaction with the enzyme. Okay, W is the difference in activation energy. So Y is the reaction with the enzyme and W is the difference in activation energy. As I mentioned, Y is, yes, the reaction with the enzyme, and W is the difference in activation energy, so yes, C is your answer. Z is the energy gained by the product. No, actually the product here loses energy. You could see how the energy for the substrate is higher than the product, so D is automatically wrong. Which statements about non-competitive inhibitors or enzyme inhibitors are correct? The inhibitor can bind to the enzyme whether or not the enzyme has its substrate bound to the active site. Is actually not true. Okay, now and non-competitive inhibitors, they bind specifically to the allosteric site. So yes, they do not bind to the place that the substrates bind to. However, when they bind to the allosteric site, the enzyme changes shape, which stops the active site from working. And as a matter of fact, I'm gonna introduce you guys to this information. The enzymes, actually most of them, they alternate between two shapes: the active shape, so this is the the active shape, and let's say this is the allosteric site, and this over here, okay, let's draw this shape. Okay, this is not the active shape, this is the inactive shape. Okay, so most enzymes actually they alternate between these two shapes. Okay. Now, what happens is that a non-competitive inhibitor is going to come in and stabilize the inactive site form. So it's only going to bind to this inactive form when there's no substrate and result in stabilizing of this inactive form. This is how non-competitive inhibitors work. You do not need to know this detail, you do not need to write this detail in the exam. However, it's just good for a question that is, I think, a little bit too tricky. So you have to understand that no, inhibitors, they don't bind with the inhibitor does not just bind when the substrate is attached. It has to bind specifically when the substrate is unattached so that it can stabilize this inactive form and change the shape of the active site. The inhibitor may bind at an allosteric site. Actually, that's the only place that non-competitive inhibitors bind. The inhibitor may not prevent substrate binding at the active site, but will prevent product formation. That is actually true. Maybe what to call the substrate does bind, but it will not maybe the substrate could, like, let's say, bind to the active site, whether or not like it's fitted for it. However, it will prevent the active product formation, because when the active site is ruined, there's no more formation of these bonds between the active site and the substrate, and so there's no more activation, or let's say basically the enzyme is not going to catalyze the reaction anymore, because it can't basically orient the substrate in the way that it needs to, and it can't provide the substrate with the things that it needs in order to do the reaction. Now, as I mentioned, again, this is one of the most tricky questions that you might find, and it's not going to get any trickier than this. Okay, so my advice to you is to just practice, because practicing alone helps you identify the trends and how the examiner wants you to answer questions. The following are all processes that allow movement into cells: phagocytosis, active transport, facilitated diffusion. Which processes require ATP? All these processes, obviously, no. Facilitated diffusion is the only process here that requires no ATP because it's a passive process. Active transport is obviously active, and so is phagocytosis because it's a kind of endocytosis 1 and 2. The fluidity of the cell surface membrane can be changed by a number of factors. As the fluidity of the cell surface membrane decreases, which process would be least affected? Okay, least affected process as the fluidity changes. Now, fluidity is going to impact things like diffusion, like osmosis, because when the membrane is more fluid, these molecules are more free to enter. But once the fluidity decreases, this thing changes. However, things that pass via protein carriers or channel proteins, these are not going to be affected by the fluidity of the membrane. They don't pass through the membrane itself. Therefore, active transport, facilitated diffusion, these are actually gonna be non-affected. So active transport, diffusion is definitely going to be affected. Endocytosis is going to also include the formation of vesicles. It also includes like things to do with the membrane, so still might be affected by this membrane change. And osmosis is the same situation, so it's really a active transport. The diagram shows the cell cycle, during which phase do chromosomes condense and become visible. When do chromosomes condense and become visible? Now, this entire thing is the cell cycle, right? So here he said this is cell division, okay? So this is cell division. So this is, I would assume, cytokinesis. Okay, A is going to be the G1 phase, then there's going to be the S phase, and then there's the G2 phase. Then here we're going to have M for mitosis, then cytokinesis is going to happen. So when do chromosomes condense and become visible? That's going to be a part of prophase, the first stage in mitosis. So D should be your answer. Now, I know this might be counterintuitive, as in like looking at this big chart to like immediately label anaphase, metaphase, prophase, and telophase, but instead we label G1, S, G2, and M, because actually this graph, like this, mainly shows the entire cell cycle, not just mitosis. Okay, so when he says cell cycle, you have to label in this way. Okay. Colchicine is a drug chemical that stops chromatids from separating during mitosis. A cell is treated with colchicine, and which stage of mitosis will this reach but not complete? So basically, we're going to like stop the chromatids from separating during mitosis, meaning anaphase is not going to even happen. So we're not going to reach anaphase. However, we're going to reach metaphase and stop there. Okay, so yes, we're not going to complete until the end in which like we transition to anaphase. I know this question is very tricky, but they tend to place questions like this, so make sure to practice as many of these situations. I think like in questions like this, you won't find many of them, but once you practice past papers, you'll be able to identify the trend as to like how to solve such tricky questions. Following statements describe the process of translation. Peptide bond forms between adjacent amino acids. Hydrogen bonds form between anticodon and the codon. mRNA binds to the ribosome. tRNA enters the ribosome carrying a specific amino acid. In which order does this process take place? Okay, so these are all the events that happen during translation. How do they happen? In what order? So the first thing that's going to happen is definitely not one. One is the peptide bond forms between it. That's the last step. Okay, what happens first is that you're going to have mRNA binding to the ribosome. Now, once mRNA is bound to the ribosome, tRNA starts to enter, and when tRNA enters with its specific amino acid, the anticodon, which is belonging to the tRNA, is going to form bonds with the codon, which means it's going to be 3, then 4, then 2. After that, when the tRNA molecules are next to each other, the adjacent amino acids are going to form bonds between one another, and so what happens is this is the last step. So 3, 4, 2, 1, which is B. Okay. Which row represents the correct features of nitrogenous bases of nitrogenous base adenine? Okay, adenine is actually a purine, so it has a double ring structure. So this is wrong. Okay, is a purine is right. It joins to its complementary base by 2, not by 3 hydrogen bonds, which means this is wrong. Pairs with thymine, that is true. It pairs with thymine in the case of DNA and with uracil in RNA. So C. Okay. The codons UGU, UGC code for amino acid cysteine, which can form disulfide bonds, and a polypeptide. The codon UGG codes for the amino acid tryptophan, which does not contain a sulfur atom. The codon UGA is a stop signal, and the DNA triplet code for the 10th amino acid in a particular polypeptide is ACA. So let's repeat this. UGU and UGC code for cysteine, which can form disulfide bonds in polypeptides, yes. And then the codon UGG codes for tryptophan, which does not contain a sulfur atom. And then UGA is a stop signal. Yes, so all these are mRNA codons. Now the DNA triplet for the 10th amino acid in a particular polypeptide is ACA, which in RNA is going to be UGU. Did he mention UGU? Yes, so this is actually cysteine, right? Yes, because it's the same codon. Okay, which single base substitutions in this triplet code will result in no disulfide bond being formed when the 10th amino acid in the polypeptide? Okay, no disulfide bond. So he said single base substitution in the triplet code. Okay, so as we mentioned, ACA is UGU and it codes for cysteine. Now mutation happens and we want to replace this, what's it called, this bond, okay, with a single base substitution. We're going to try to replace this amino acid with another one. He said tryptophan does not form sulfur atoms. So what happens to ACA is it needs to change to UGG. Now UGG stands here for A. Now you're going to have to translate this ACC. So another way to convert to ACC is simply to change the C and the A here. Okay, so ACC and ACG. Okay, does ACG also code for cysteine, which can form a disulfide bond? Or we could also convert it to UGA, which is ACT. Okay, so ACT. So we could also change it with T. So it's either ACT or ACC, which is C. Okay, I hope this is understandable. Let me repeat this really quickly. He said here that we have specific codons for specific amino acids, UGU, UGC being for cysteine, UGG for tryptophan, UGA is a stop signal, and he said that we have a 10th code for what's it called, a DNA triplet code for the 10th amino acid in a particular polypeptide is ACA. Now this ACA, if you convert it to a codon, it's UGU. It's actually cysteine. It's listed here, okay, as UGU. Now he said that he does not want if let's say a mutation happens and we have a single base substitution in this triplet, and so instead we have no disulfide bond. What could cause this to, what would basically be the substitution that would result in no disulfide bonds being formed? Well, obviously one that results in an amino acid or a stop signal that basically do not form disulfide bonds. And here he gave us an example of this, which is tryptophan, which is UGG. So we convert this to a DNA code and figure out how this change happens, such that cysteine turns into either a stop signal or into what's it called tryptophan amino acid. Okay. A student produced a table of differences between RNA and DNA. The table contains one mistake. Which row has the mistake? RNA and DNA, and so there's a mistake in the row. RNA contains ribose is true. DNA contains deoxyribose is true. Contains uracil is true. Contains thymine is true. Found throughout the cell. Yes, RNA is found throughout the cell. Found only in the nucleus is actually wrong. Okay, because DNA is found also in the mitochondria in circular form. So no, it's not found only in the nucleus. Okay, single polypeptide per molecule. A single polynucleotide per molecule, that is true, and then 2 polynucleotides per molecule is true as well. So C is your. The diagram shows a transverse section of parts of a plant with some transport tissues labeled 1 through 6. So you have 1, 2, 3, 4, 5, 6. Now, here, so we can label 1 as the phloem, and 2 as the xylem, 3 as the phloem, 4 as the xylem. Okay, let me write, let's let me write X for xylem and P for phloem, and then here we have X and P, and then here we have again X, and this is P. Now it says which tissues mainly transport water and which tissues mainly transport sucrose. Okay, mainly transport water, that will be xylem. Mainly transport sucrose would be phloem. So 1 and 5 transport water. 1 and 5, 5 is a phloem, so this is wrong. Okay. 2 and 3. 2 and 3. 2 is the what's it called, phloem, and 3 is also the phloem, so this is definitely wrong. Okay, 3 and 5, as mentioned, 3 is the phloem, so this is wrong. Okay, 1 and 6, and 1 and 6 is right. Okay, and then 2 and 3 for phloem. Yes, fantastic. So this answer would be D. The transpiration is the consequence of which features of plants? Okay, which features of plants? The presence of symplast and oplast pathways? No. The requirement of transport system in plants? Well, that's definitely a consequence of having transport systems. The requirement for leaves to exchange gases. The thin waxy cuticle on the lower epidermis of the gases. You're gonna have to select C for this one. Okay, transpiration is going to happen whenever water is going to leave through the stomata. Now, obviously, water is going to flow all the way from the roots through what's called xylem into your leaves, and then from the leaves through the stomata into the air. That will be transpiration. Now, yes, the requirement of transport system in plants, it could maybe result in transpiration, but it's not until what's called plants have stomata and are allowed to do gas exchange that transpiration can actually happen, which is why I'm selecting C. The thin waxy cuticle on the lower epidermis of leaves, waxy cuticle kind of as a thing that actually reduces the rate of transpiration. So really, you would have to say that it's actually something that stops transpiration, not results in it. So D could be cancelled. 24. Mass flow is the bulk movement of materials from one place to another. How many of the vessels listed carry fluids by mass flow? Okay, by mass flow, the artery, the phloem, the vein, the xylem. Okay, so actually, in this case, arteries and phloem and veins and xylem vessels, they all carry fluids by mass flow. Okay, mass flow is like the flow of fluids all in one together. Now, I can't definitely remember the definition, but I'm going to write it down here for you. Okay, so this is D. The diagram shows a xerophytic leaf in different conditions P and Q. So you have X and X. Which statements describe the difference between the cells in layer X in conditions P and Q? Okay. So you could see how P is, so let's see. Like, he's looking for specifically this difference in cells in layer X in conditions P and Q. Now, X cells are definitely smaller. It's the reason the whole thing is curved up. It's because they all shrank together. Versus if you look at Q, they're all turgid, they're all flat, and so this is why it's bigger. And so obviously the difference in conditions is that there's a lot more there's a lot less water in these cells than in Q. So there's a more negative water potential in P than in Q. Okay. More negative water potential in P than in Q. Now, what it means to be more negative is that it's more concentrated. Okay, and yes, when you have less water, it's going to be a more concentrated situation. So yes, it's more negative water potential. More cells are plasmolized in P. Again, it's another situation that could happen because these cells are in such maybe hypertonic conditions, and therefore these cells are very shrinked. Cells are less turgid in Q. Actually, that's definitely wrong. Cells are more turgid in Q. Water potential becomes 0 in Q. Water potential becoming 0 in Q is not necessarily 100% true. Like, we don't know if the water potential is exactly 0 and that it's placed specifically in distilled water, we just know that it's placed such that it's turgid, it's fine. Okay, so it's actually going to be 1 and 2 only, which is B. Okay. What could increase the rate of transpiration? Increasing humidity, increasing light intensity, decreasing temperature, decreasing the wind speed. What you will want to do is you increase light intensity, because when you increase light intensity, plants do more photosynthesis, they do more gas exchange, so there's more stomata open, so there's a greater rate of transpiration. Decreasing temperature is going to decrease the kinetic energy of water molecules, decreasing the rate of transpiration. Decreasing wind speed is going to decrease the diffusion gradient, and so B is your answer. Okay, what happens during ventricular systole in a mammalian heart? During ventricular systole, is it that aortic pressure increases, atrioventricular valves open, semilunar valves close, or ventricular pressure decreases? Well, what happens during ventricular systole is that the ventricles are going to contract, which means that the atrioventricular valves, so let's draw the heart. I'm not sure if this is a good idea or bad. I think it's a bad idea. Okay. So let's imagine that this is a heart, please, okay? So this heart over here, okay, is going to have a bunch of blood vessels coming in, coming out, and then same situation here, going in, and then going out. Okay, I tried my best, guys. Anyway, what happens here is that the ventricles are going to contract, and when these ventricles contract, the pressure inside them is going to really, really increase. Now, to avoid blood flowing backwards into the atria, what happens is that these valves over here, the atrioventricular valves, they're going to close. So the atrioventricular valves are going to close. Meanwhile, we're trying to push blood out of the heart, so out of the heart is where the blood wants to go, so meaning we need to open the semilunar valves. So semilunar valves are going to open, so this is wrong. Okay, now when semilunar valves open, pressure in the arteries is going to vastly increase. So yes, aortic pressure increases. Atrioventricular valves open is wrong, and ventricular pressure decreases is wrong, therefore A is your answer. Okay. Which substances could displace oxygen from oxyhemoglobin? Displacing oxygen from oxyhemoglobin, something that has a higher affinity to what's it called, to hemoglobin, so carbon dioxide could definitely displace, carbon monoxide could also displace, hydrogen carbonate ions do not displace it, hydrogen ions could also displace, but it depends again on affinity, which have more affinity, who does hemoglobin have the most affinity to? Hemoglobin's greatest affinity is going to be to carbon monoxide, and then after that to hydrogen ions, after that to oxygen, after that to carbon dioxide, which means your answer is going to only be hydrogen ions and carbon monoxide. Obviously, that all the carbon dioxide can displace oxyhemoglobin to form carboxyhemoglobin. That's only when carbon dioxide is in really, really high concentration. Okay, so it's going to be 2 and 4, which is D. The graph shows the affinity of hemoglobin for oxygen at 2 different concentrations of carbon dioxide. Which point on the graph shows the percentage saturation of hemoglobin entering the pulmonary artery? So hemoglobin is going to enter the pulmonary artery. And so in the pulmonary artery, hemoglobin is going to be deoxygenated, which means it has a obviously a lower affinity to, so percentage saturation of hemoglobin with oxygen is going to be lower, and it's going to have higher carbon dioxide or low carbon dioxide is obviously it's going to have higher carbon dioxide. So entering the pulmonary artery, right? It says entering the pulmonary artery. Okay, entering the pulmonary artery is going to be just as deoxygenated as as the blood flowing in the vena cava, right? So the percentage saturation of hemoglobin with oxygen is going to be quite low. It's going to have really high carbon dioxide, and the partial pressure of oxygen is going to be also quite low, which means the answer here is going to be B. Okay, the diagram shows some of the reactions of carbon dioxide when it enters blood from cells in a metabolically active tissue. Which reaction is catalyzed by the enzyme carbonic anhydrase? Okay, the enzyme carbonic anhydrase catalyzes the formation of carbonic acid from carbon dioxide and H2O, which is which one? Is it CO2 reacting with H2O and forming this in the plasma, or is it found specifically in the red blood cell? It's obviously found in the red blood cell. Okay. How many phospholipid bilayers does oxygen pass through in diffusing from the alveolar air space to form oxyhemoglobin in the red blood cell in a mammalian lung? So from alveolar air to the red blood cell, okay, how many plasma membranes are we going to pass through? This is a terrible attempt at the red blood cell, and then this is the, the capillary, and then this is the alveolus formed from, what's it called, single cell squamous epithelial cells. Okay, now the squamous epithelial cell is going to have two membranes, that's 1 and 2, and then so that's here 1, 2, and then it's going to pass. And now he said, how many phospholipids does oxygen pass through in diffusing from alveolar air space to oxyhemoglobin in a red blood cell in a mammalian lung? Now, how many does it pass through? Okay, so not necessarily how a maximum or minimum number, but what's it called? Let's assume it passed through the capillary walls. So 3 and 4 and 5, and 5 is it? Okay, so it's 5. Okay. Now, what's it called? Which statements about the gas exchange system are correct? Which statement was okay about the gas exchange? Absence of cartilage and small bronchioles allows them to expand. The absence of cartilage and small bronchioles allows them to expand. The walls of the alveoli are made of cuboidal epithelium. They're made of squamous epithelium, so 2 is wrong. Okay, the alveoli secrete surfactant, which reduces surface tension in the lungs. The alveoli do secrete surfactant. This is supposed to be a material that's going to decrease surface tension, allow the alveoli to be held in place as they are. And the trachea and bronchi are supported by circles of cartilage. Trachea is supported by circles of cartilage, but for bronchi it's actually supported by blocks of cartilage. So really the only right answers here are 1 and 3. Okay, so that's B. I don't think I explained 1. The absence of cartilage in small bronchioles allows them to expand. As you move on from trachea to bronchi to bronchioles, cartilage is no longer there. Now, cartilage is there in order to support these things. Now, in bronchioles, because they're so small, they don't really need to be supported, and so yes, they're able to expand when they're trying to when we inhale air, they expand because there's no cartilage. Now, a disease damages alveoli walls, causing alveoli to burst. What effect does this have on the gas exchange system? Surface area for gas exchange is definitely going to decrease because when you have less alveoli, you have less surface for gas exchange. Maximum volume of air breathed out. What's going to happen to the maximum volume of air breathed out? Now, there's going to be a decrease in the surface area. So let's imagine what's going to happen. You have less alveoli than before. Okay, so these are the alveoli. Now, usually carbon is going to enter, oxygen is going to leave, okay, and, this is going to go out just fine. But then, let's say some of these guys are going to burst, so we only have 1 left. Okay, there's going to be only this, the blood from here providing it with the carbon dioxide. So I think that, yes, it's going to have an overall decrease. So A is my answer. Okay. What are the short-term effects of nicotine on the cardiovascular system? Nicotine on the cardiovascular system is not going to be in the syllabus, therefore we can cancel question 34. What is correct for tuberculosis? So tuberculosis is an airborne transmission. That is true. Is it caused by a virus? No, it's caused by a bacterium. Does it cause rashes? No, rashes is not one of the symptoms of tuberculosis. So A is your answer. Factors contribute to outbreaks of measles after natural disasters. Contamination of drinking water with untreated sewage is actually going to contribute maybe to the factors that lead to outbreaks of maybe cholera. Not to measles, because measles is an airborne infection. Lack of effective vaccination coverage in the population before the disaster. Exactly. If you have lack of vaccination coverage, there's a lack of immunity within the population. This is going to lead to an outbreak. Okay, people living in overcrowded accommodation. True, because as I mentioned, it's an airborne infection. Once someone gets the disease, there's a big fat chance that everyone else is also going to get it if they live in a very tight situation. Which of these statements explain why some vaccines can be taken by mouth, but tuberculosis vaccine has to be injected? And now the tuberculosis vaccine could be generally it's going to be digested in the gastrointestinal tract. Therefore, it's not going to be taken via the mouth. Just like actually most vaccines are injected. Macrophages present antigens and vaccines to stimulate an immune response. Macrophages present antigens and vaccines to stimulate an immune response. This is not the reason that they're taken by mouth. Okay, what happens is that vaccines are going to be given to you. They're going to be a weakened version of the disease, and then the immune system is going to fight it. So no, okay, it's not one. The TB antigens necessary to produce an immune response are proteins which would be digested in the stomach and the small intestine. Exactly. There are no B lymphocytes and T lymphocytes in the stomach is not the reason. There are specialized B lymphocytes and T lymphocytes in the stomach. Therefore, no, it's just going to be 2. Okay. Which method of gaining immunity can be described as natural active immunity? Okay, natural active, feeding on colostrum. Colostrum does provide antibodies, however, it is a passive immunity, so no. Inhaling the chickenpox virus. Inhaling chickenpox virus. Well, chickenpox virus is it's definitely natural active immunity. Okay, so yes, because you get so natural active immunity, the only way to get it is basically to get the disease yourself firsthand, deal with it, and obviously now you have the immunity, you have the memory cells, you have the antibodies, you have everything. Okay, injection with antibodies is a passive artificial immunity. Through the placenta is a natural passive, again, so it's B. Which statements about endocytosis are correct? Okay, about endocytosis. It's a part of phagocytosis is true. It's a passive process is wrong. Of course, not. Endocytosis is an active process. It's basically going to have, the membranes are going to close up and they're going to form vesicles. And so, no, the process requires a whole lot of energy, actually. Okay, for the membrane to move like this and for like the motor proteins to like pull the membrane like this together, it's going to require a lot of energy. Materials are taken into the cell. That is true. Okay, vesicles form within the cytoplasm. Also true. 1, 3, and form 4. Okay. The statements refer to vaccination programs for each of the diseases: cholera, cholera, measles, smallpox, and tuberculosis, TB. Which statement is correct for TB? Most children are vaccinated before their first birthday. Actually, I don't think so. I don't think most children are vaccinated before their first birthday. It's usually before. It's usually like in. Like if a child was to be vaccinated, because not everyone is vaccinated for TB. It depends on like where you live and like the conditions in which you live. So no, usually they're not vaccinated before their first birthday. The pathogen has not been mutated or changed its antigen. Actually, no, there's a bunch of mutations to TB to this day. Pathogen lives in the intestines where antibodies cannot get to it. That is not true. It does not live in the intestines. Okay, it usually actually lives in, I think, the lymph nodes or in the lung. In the lungs. Okay, the vaccine is not effective against some strains of pathogen. That is true. There is a vaccine known as the BCG, the Bacillus calmette-guerin. It is going to be specifically for Mycobacterium bovis, but Mycobacterium tuberculosis is not going to be effective. The vaccine is not going to be effective for this strain of tuberculosis. So this is true. D. Okay, so this is it for this video. I hope this video was very useful to you guys, and if it was, make sure to please like, share, and subscribe to this channel. Now this is it. I'm gonna see you guys in the next one.